Ammonia is toxic in high concentrations, and so it must be incorporated into biologically useful compounds when it is formed by the reaction of nitrogen fixation, discussed in an earlier lesson. The amino acids glutamate and glutamine are of central importance in the process. Let's take a look at that process and get an idea of the patterns observed in amino acid biosynthesis. There are two observed reactions that incorporate ammonium ion to form amino acids. The first reaction shown below is the formation of glutamate from the reductive amination of alpha-ketoglutarate. Note that this is an amination because it results in the formation of an alpha-amine group and costs one NADPH molecule. The second reaction involves the amidation of glutamate, which we examined in the previous lesson. Note that this is referred to as an amidation since it results in the formation of the amide side chain of glutamine. Once incorporated into one of these two amino carriers, the most common means of synthesizing other amino acids is by a process known as transamination, the transfer of amino groups between molecules, which we will examine in detail in the next lesson. Since these biosynthetic pathways are very detailed, we want to focus on the main principles and general schemes. For instance, we can group amino acid biosynthetic pathways into families, as illustrated here. In other words, several amino acids might be generated by a common precursor. Some of those amino acids are then used to generate others. Although this was referred to in an earlier lesson, we will now see more particularly how citric acid cycle intermediates are key intermediates in the synthesis of amino acids, as demonstrated in this diagram. We will also find that amino acids can be broken down into TCA cycle intermediates, as illustrated here. Thus, the citric acid cycle is truly amphibolic, related to both anabolic and catabolic pathways.